Okay, so our next presenter is also a recipient of the Baer Foundation Prize and is also a member of Dr. Caron's lab at Duke and is also an international person. He's a senior postdoctoral fellow at Duke. His name is Nikhil. He did his undergraduate and master's education at the University of Mumbai in India, and then came to Atlanta to Georgia Tech. Was that the same thing? Georgia Tech is the same thing as, yeah, okay. As Georgia Institute of Technology, where he pursued his doctoral studies, and he joined Dr. Caron's group in 2007. He's been working with genetically altered mice to explore and understand better how antipsychotic medications work, their mechanisms of action, as part of some of the things we've already heard today. So, Dr. Urs, please join us.
Hello, good morning everyone, and thank you for being here. And thank you, Dr. Hirschfield, for that very kind introduction. And so I would like to thank the Baer Foundation for this award, and both the Baer Foundation and the BBRF for providing this platform to allow researchers like us to show you what we're doing. And so today I'm going to talk about a little bit of what we are doing in the lab, trying to develop functionally selective drugs as antipsychotics and try to understand the mechanisms behind schizophrenia. So why do we study schizophrenia in animal models? So as a basic research scientist, we're not privileged, or rather we're not fortunate to see patients in the clinic or look at people with mental disabilities. So what we do have is all this data with human clinical studies that we can use, and we can apply these data to develop better mouse models that we have in the lab. So what is traditionally being done is that you have these antipsychotics that are currently being used in the clinic, such as risperidone or olanzapine. And as Dr. Zurasa mentioned, that you cannot ask a mouse whether it's having hallucinations or delusions, but there are certain behavioral outcomes in these animal models that are close correlates of the symptoms of schizophrenia. And what is traditionally being done is that these behavioral outcomes are being studied in mouse models, and researchers test the effects of antipsychotics on these behavioral outcomes, and that allows us to develop better medications. And what is recently being developed is these pathway-specific animal models. So when biochemists and molecular biologists look at these different specific signaling pathways that Dr. Caron talked about, what they can now do is develop pathway-specific animal models, and this could lead to the development of pathway-specific therapeutic drugs. So what we are doing in the lab right now is trying to identify the role of different cell types using these mouse models. So as I told you, that we can use the data from human clinical studies, which tell us that there are these different brain regions that are very important in the manifestation of the symptoms of schizophrenia. So we can look at those same brain regions in the mouse models that we have in our laboratory. And so just to give you an example, so we have been using these Cree lions that Dr. Caron mentioned, and these were developed by the GenSat consortium at the Rockefeller University. And what this allows us is to delete specific genes in specific cell types. So for example, you can have a mouse model where you delete your favorite gene in this brain region or this brain region here. And we know that both the cell types here are totally different. And then you can also have another animal model where you can delete your gene of interest in just one region, but not the other region. So what this does is now you have these two different animal models, and you can compare the behavioral outcomes in these two models, and then you can figure out what does this brain region do or what does this brain region do, depending on the behavioral outcome. And so using these animal models, what we have identified is that when you delete a certain gene of interest in one particular region, you get a behavioral deficit. Whereas if you delete the same gene in a different region, you do not get the behavioral deficit, suggesting that one particular brain region is responsible for this behavioral outcome in this animal model. And so using these animal models and this technology, what we have been able to show is that drugs like antipsychotics and other drugs like lithium that is being used for bipolar disorder treatments have some common mechanisms, the way they work, but there are some mechanisms that are totally different, and that's why antipsychotics are used for schizophrenia, whereas lithium is used more for bipolar disorder. And what this does is it gives us an insight into how these drugs work and how we can develop better therapeutic options. But, we are looking at specific gene knockouts in different cell types, but the big question is, what is happening in the whole cell? What is happening to all these cells here? What is happening to all these cells here? The cell consists of thousands of proteins. So the big question is what is happening to these proteins in these different cell types? And so what we are essentially developing in the lab right now is this barcode for protein profiles in different cell types. So you can imagine that you have a certain cell type in this brain region and a different cell type in this brain region here. So we have this technology where we can specifically label proteins, all the proteins in a particular cell type, and then use a proteomic approach where we can identify what kind of proteins are being changed in these cell types versus these cell types here. And now we have this transgenic mouse model in our lab where we can actually create this barcode for all the proteins in these cells and essentially try to figure out what is the difference between this cell type here and this cell type here just based on their protein profiles. But the big question is, what would happen to these barcodes in a normal mouse versus a mouse that shows some of the symptoms of schizophrenia? And so you can imagine that once we use this proteomic approach, we can get a protein barcode profile for a normal mouse and a completely different barcode profile for a schizophrenia-like mouse. And then we compare these two mouse lines and we see what are the differences in their protein barcodes. And let's say we identify, like Dr. Caron was saying, like the silver bullet or maybe a couple of bullets, or an entire magazine, who knows. But let's say you identify these proteins and the differences between a normal mouse and a schizophrenia-like mouse, then what do? We get new targets, and we can now generate functionally selective therapies against these newly identified targets. And so in conclusion, our big goal is to use in vivo animal models and use transcriptomics like Dr. Caron mentioned, and use this newly developed proteomic approach where we can get this protein barcode profile and try to identify novel targets in these mouse models, and then go into the lab and try to identify better therapeutic options to treat these behavioral outcomes. And then the hope is to then generate some functionally selective or biased drugs which can then go towards clinical testing. So with that, I would like to end the talk, and if you have any questions, thank you.
Okay, if we can get the lights up again then, please, and questions. Actually, I'd like to ask you the same question I asked Dr. Caron. can this technique, especially this proteomic technique, lead to personalized medicine in some way with our illnesses?
Yeah, definitely. I mean, once we have access to the protein profiles in the different cell types, and using the human clinical study data, if we can develop different mouse models based on different behavioral outcomes, then we can develop different protein profiles for each behavioral outcome, and hopefully that could lead to personalized medicine.
That's really exciting. Questions for Dr. Urs? Oh, yes, sir.
And my concern is we keep talking about all these different communication channels that are there, all right? And they work most of the time. And they're composed of different protons, all these different things. Perhaps we're looking the wrong way. Instead of looking for bugs, little tiny protons that aren't working, perhaps we should be looking for the insulator of these wires. Maybe that is where the problem is, because the problem is all over the brain in different spots with different kinds of genes involved. Maybe it's the insulators that aren't working. You know, we have a pack of wires with an insulator. It isn't there.
Yeah, that actually is that's an excellent comment though. And actually that is what we are trying to look for in the future. Once we know what's happening at the cell level and at the individual protein level, then we can identify the circuits that are going wrong in a normal mouse versus a mouse that has a schizophrenia-like symptoms. And then we can compare these two and identify what kind of circuits are altered. And I guess that's what you're trying to get at as the holistic, what's going on in the whole brain.
So what I'm more concerned saying is the fact that the silver bullet might be found in the insulator. It doesn't matter what is the different wires or whatever. The wires matter, but it's the insulator that's causing the problem. Because we're having different problems, one of the problems, but we have different problems in different parts of the brain. It might be just as simple as
This articulates nicely with an earlier question about someone who had MS, which is a disease of conductivity and schizophrenia, I think it was. So that's a very interesting concept. Other questions for Dr. Urs? Yes, way over here. Wait, right here, this way? Couple.
You talked about the barcode,
Right.
and would this be a way to not only target the positive symptoms of schizophrenia, but also the other symptoms that are currently not being targeted by antipsychotics?
Right, that's exactly what we are trying to do is like, once we have the right animal models that display, let's say, the positive symptoms of schizophrenia, and there's another mouse model that displays only the negative symptoms of schizophrenia, then we can use those mouse models to look at what are the protein barcodes in those mouse models, and that will allow us to identify new targets that were previously unappreciated, and maybe we could, finally identify something that would treat negative symptoms. Yeah. Oh, right there.
Okay, last question here. Yeah.
I would like to ask a very simple question.
Yes, please.
You say that, and we know, mice cannot tell us their thoughts, et cetera, et cetera. So what are the characteristics that you can induce in a mouse that is similar to schizophrenia? You say schizophrenia-like symptoms, such as what?
Yeah, so there are a few examples, like drugs like amphetamine or PCP, when they're given to patients, they exacerbate the symptoms of schizophrenia, like in humans. So when you use these same drugs and you give them to mice, they induce like a hyperactivity-like state, which is a loose correlation of what you would see, or I would say a manifestation of that kind of behavior induced by those drugs. So that is one of the behavioral outcomes that we are using in these mouse models.
Is another one being antisocial? I mean, do mice generally stick together?
Yeah, that's one of the pictures that Dr. Caron had shown before, that in these mice that are lacking one of the glutamate subunits of the glutamate receptor ion channel, is that these mice Usually mice, they bundle up and they always sit in one corner, but these mice, they all stay separate, and so that's actually a very good model for the negative symptoms of schizophrenia. And the idea is to develop like this protein barcode profile in these different animal models so that we can identify what's happening in these mice which are displaying negative symptoms versus mice that are displaying positive symptoms. Yeah, thank you.
Thank you. Thank you, Dr.
Thank you.